I'd like to thank our president, Dr. Alba, and also Summer Professor Marces Sondro. Okay, today I would like to talk on evolving concept of genetic diversity. In particular, as our chairman kindly introduced me as genomics, so I would like to consider evolving concept of genetic diversity from genomic evolution, particularly neural system, and also I'm going to talk on eyes. But before that, I would like to show some our recent achievement. This is sushi, this is a tuna, and last year we sequenced complete genome of Pacific bluefin tuna. It's very interesting. I won't go into detail. And this is maki sushi. And also we sequenced complete genome of sushi nori, red algae. Also very interesting. But anyway, such a genome technology is advanced. And what I would like to introduce this technology just this year, actually like it started so many years ago. It is USB sequencing, just a USB type. And using PC batteries, you can sequence human genome only 2.5 hours. So if you have like 10,000 PCs, you can sequence 10,000 human genome within 2.5 hours. I would say you don't need any big genome center. It's such a tremendous advancement is taking place. And since I'm interested in neural system evolution, I'd like to review what we have been done so far. Then I would like to go into evolution of eyes, particularly taking into account evolving concept of genetic diversity. And the first question was, what kind of genes are expressed in a planarian brain, which is supposed to be the most primitive brain? Planarian is very small primitive organisms which has strong regeneration power. And this is kind of phylogenetic tree. Sorry, I don't know how I can point out. But anyway, the chordata, it's the lowest one is a human, the vertebrate. But if you go further, just yellow point shows planarian, which seems to have just emerged having central nervous system. Because when you look at comparison between a very small planarian brain versus human brain, size is so much different. However, this planarian species has two eyeballs and optic chiasma, and just like a human. Also, one specific gene, which are well known to be specifically expressed brain, OCT, OCT gene, has been expressed in a planarian like those organs. But when we regenerated completely identical, genetically identical clones, more than 500 individuals, then cut off the head and compared head part and body part. Then we conducted so-called in situ hybridization, which means by markers, you know where those genes are expressed in planarian body. What we found, it's a tremendous different gene expression patterns. And also we found kind of regional functionalization. Already planarian brain seems to have a different region depending on function according to gene expression pattern. So functional regionalization of planarian brain, also which means kind of ancestral genes which seems to be sharing with human brain genes has been already expressed even like 700 million years ago. Then next question, we went into much more primitive organisms. That is called Hydra, Cnidarian, which is known not to have a central nervous system. What just beyond Planarian, according to phylogenetic tree. And this Hydra is very, again, small, like a half inch long, and it has a tentacles, and it is like stabilized the soil under the seawater. As you see, kind of a dot over the body, that's the neural cells. Some cells called nematocytes, which can control like a movement and catching bait. And we wanted to know what kind of genes expressed in those neural cells. But unfortunately, different from Planarian, they don't have any central nervous system. Therefore, we have to have something. Otherwise, we cannot isolate genes from particular single neural cells. Fortunately, we have mutants. That mutant is called epithelial hydra, epihydra, which lacks only neural cells and nematocytes, which cannot move at all, but you force them to eat, then they can survive. So by comparing those normal hydra and the epihydra, we can check what kind of genes are expressed in normal neural cells. Then we found it. We checked gene expression patterns by making markers. Again, a lot of interesting pattern, which means even though Hydra may not have a central nervous system, still they have a tremendous integrity to regulate its own behaviors, maybe kind of metabolism, and having a gene regulation system. So, genome regulation network already exists in human neural cells, even though they do not have central nervous system. Up to here, this is a kind of a review what we have done. Then this is a kind of a new issue. Then just going back to a little bit higher organisms, I went first like a Planarian, then much older organism, Hydra, but then coming back, sea urchin. Sea urchin is much higher organisms than Planarian and Hydra. As you can see, it looks central nervous system emerged just before Planarian emergence. But sea urchin does not seem to have central nervous system, apparent central nervous system, or apparent so-called ganglion. Therefore, from this phylogenetic tree, the sea urchin lost their central nervous system, at least ganglion. Then what happened? Sorry, this is a loss of central nervous system because sea urchin is after emergence of central nervous system. If you look at the left side of your screen, green means it's an adult form. It's completely, I would say, a radial form, symmetrical. And according to protein markers, which does not show kind of a central nervous system, only they have like 5 spots. But the right side, it's a red picture shows larval form. And the larval form is not symmetrical in a way. And this is a kind of this form, two just like antennas and this form. If there is a ganglions or central nervous system exist, one possibility is here, so-called apical organ. What we have done is now we prepared genetically identical sea urchin individual, almost 300 samples, then cut off that region, then we checked gene expression patterns. Also, we have to note that complete genome of sea urchin has been already known. Since we know complete genome, what kind of genes are encoded, but probably or possibly there might be that some particular genes which are specific to, say, central nervous system may not be expressed. So when we sequence a lot, then making annotation, then we found genes exist in the genome, but almost 2/3 of genes which are seemingly related to nervous system are not expressed. Therefore, maybe kind of a loss of gene expression is causing a loss of like a ganglion in the sea urchin. So just like apical organ there. But interestingly, if you just, upside down, just like a human, almost the same region. So from here, probably the sea urchin used to have a central nervous system, but somehow because of loss of gene expression, they might have lost central nervous system. Remains of brain in sea urchin. Okay, then we wanted to see like eyes, camera eyes, because of course what we really want to know is connection between such a sensory organ and brain system, but it's so difficult. Therefore, first we wanted to see evolutionary process of camera type eye. This is a human eye, but from the time of Charles Darwin, it is known such a vast amount of different eye, pinhole eyes, complex eyes, and like mirror eyes. And it is known that almost 6 major different types of eyes. According to Charles Darwin, in his famous book on origin of species, he suffered from understanding of diversity of eye. Such a tremendous, complexity and advancement, and how it can be explained by natural selection. And he spent one chapter, but concluding that still he believed that this can be also explained by natural selection. But anyway, according to Walter Gehring, who unfortunately happened to pass away by a traffic accident last May, he made a hypothesis all eyes may have originated one single ancestral form. At the time of Darwin, even Darwin speculated that eye might have emerged independently at least 40 times. But according to Walter Gehring, he said that it could be monophyletic origin because of his PAX6 gene studies. And in particular, the Nautilus, this is a deep sea seashell, has a pinhole eye. Scallop has mirror eye. Octopus has camera type eye. Particularly, we focused on octopus because which has a camera type eye. When you compare the human eye, octopus eye, very similar. However, because of characteristics, like a human has nervous cord at the surface, but the octopus has it in the backside, and the human focus changing thickness of lens, but the octopus focuses only the location of lens. Also, like proteins made of human lens is alpha heat shock protein mostly, but the octopus lens is made of completely different protein. From these observations, evolutionarily, those two eyeballs are completely independent origin, which we call convergent evolution. Origins are different, but the structure functions are quite similar. On the other hand, if it is monophyletic origin, it is called divergent evolution on the comparing with convergent evolution. So, convergent evolution or divergent evolution. So we took octopus and isolating eyes, constructing cDNA libraries, and sequenced. And we obtained a lot of sequence, but only what we can successfully made annotation was 1,052 genes, which seems to be related to kind of functions, including not only eye functions, but also other, regular cell function. So question is, out of 1,052 genes, how many genes are shared with genes expressed in human eyes? If percentage would be more than 70%, 80%, it could be divergent evolution because monophyletic sharing common ancestor. On the other hand, maybe sharing percentage is only 20 or 30%, which shows that just genes which are necessary to maintain a basic cell function. Therefore, it's already common, but others could be different. So therefore, question is 70 or 80% of sharing, or only 20 or 30% of sharing. So fortunately, there is a human eye gene bank. So out of 1,052 genes, how many genes are shared with human eye genes to be expressed? The answer was about 70%. So from here, we speculate. Of course, definition of convergent evolution is so difficult. But still, we say, according to common knowledge, it could be divergent evolution instead of convergent evolution. Okay. So we went into a little bit more. Then what is the evolutionary origin of eyes? Actually, when you look at seawater and a lot of planktons, but the one of plankton, it is called dinoflagellate. In the world, it is known that more than 2,000 different species exist. In particular, Warnobiae, this dinoflagellate is so interesting. This dinoflagellate is a single-cell eukaryote, but in particular, Warnobiae has this one, just this type. Different dinoflagellate has different photorecepting, possibly. We don't know the photoreceptor or not, but the photorecepting probably kind of organ. However, dinoflagellate, this Warnobiae has very similar lens-like camera lens-like structure. And three different species share almost the same structure, which seems to have a lens, also retina-like structure, even though this is a single-cell eukaryote system. Just like this. The upper part seems to be a lens, and the longer part seems to be a retina type. And when we check dark and the light conditions repeatedly, the structure changes. Apparently, they are recepting the light. And moreover, when we isolated DNAs, then we found rhodopsin. And more interestingly, this rhodopsin is not eukaryotic rhodopsin. This rhodopsin is bacterial type rhodopsin. Therefore, those genes seems to be horizontally transferred from bacteria, possibly, because dinoflagellate has a lot of symbiont of cyanobacteria. Also, dinoflagellate itself has a symbiont coral or jellyfish. And even we found within the structure DNAs, which are different from its own, the host DNA. Moreover, we found this structure replicate itself within the cell. So from here, we strongly believe this could be like organella, such as mitochondria. Therefore, most probably, this organ seems to be horizontally transferred. Okay. So Walter Gehring said that the Russian doll models, which means that the cyanobacteria horizontally transferred to dinoflagellate, dinoflagellate possibly transferred to jellyfish. We are now checking the genome of jellyfish, and somehow those becomes our eye system, which we have to prove. But such kind of horizontal genome transfer may have significant importance during evolution of eyes. This is still hypothetical situation. Cyanobacteria. So, genes of eye structure, particularly in dinoflagellate, were horizontally transferred. From here, we conducted massive operation of searching marine plankton, if they have kind of genes which are related to neural system or not. So we call marine micrometagenomics. Metagenomics is just you take a seawater samples, you don't care about the culture ball, or you cannot see microscopic features, we don't care. Just sequence immediately. Then what kind of gene or genomic segment are, check the database, then we know what kind of microorganismic compositions are. So particularly like this is a Japan island and the ship. This is a kind of a cylinder taking out seawater. Right now, our laboratory has advanced technology. We can use only 1 liter seawater. At the time when Dr. Craig Venter had seawater samples, metagenomics, it needed 400 liters, but we need only 1 liter. So then filtering on the boat, and in particular, we are interested in Sendai Bay, where a huge earthquake hit. Then how ecological changes, particularly as the microorganismic diversity has been changed. Also as a control, we take the samples, also the sea samples has been reserved off Hokkaido seashore, which seems not, does not seem to be affected big earthquake. Anyway, the comparison itself has not been made yet, therefore we don't know. But anyway, also we took the sea samples from Kyushu Island. Then we really developed isolating like DNAs from seawater, depending on the size, we can take planktons, now bacteria, even marine viruses. And amazingly, we are obtaining a huge amount of marine viruses that we never be able to see. However, apparently, a lot of DNAs are obtained. We have not sequenced viruses yet, but so far we sequenced 110 billion nucleotides only by our own project. Such a huge nucleotide sequence data gives us how much horizontal gene transfer is taking place at this seawater regions, and the bacteria versus planktons, bacteria versus bacteria, others. And just showing some result, and now we can make kind of the phylogenetic classification. And the conclusion is, most cases, one dominant bacteria, say 70%, 80%, but the other major bacterial species only 10, at most 20. Then 300, 400, those bacteria are a small amount, such a very much characteristics. Also, what we found is it does not matter where you take the samples, like 100 kilometers away, almost the same. However, it changes dramatically because of season, and most probably seawater. Therefore, tide current, maybe kind of a microenvironment changes does not matter much. However, such a seasonal change makes a tremendous contribution to the change of microorganismic composition. Okay, just like this. So even we checked red tide. Red tide is very much important phenomena for aquaculturing industry. If red tide comes and the most fish and agricultural organisms die. But when we check the bacterial composition, a few number of particular bacteria comes up just before red tides. Therefore, those microorganismic diversity, by looking at DNA markers, can be a good caution or prediction of red tides. Therefore, industrial application also possible. We are not so much interested in industrial application, but still it's possible. Right now, we are developing big buoy. And here, automatically we can take samples. We don't have to go to the sea by ship. And this means such kind of metagenomics can be made monitoring system, automatic monitoring system. Still, we cannot place the sequencing on the buoy. However, as I said, that if USB sequencing is possible, even sequencing can be done automatically on spot. And we have already 3 type of this buoy which we made, and now still advancing. And also, just last year, I had a laboratory in Saudi Arabia, King Abdullah University of Science and Technology. Fortunately, they have a Red Sea. And the Red Sea has a tremendous high concentration of salt. And because of that, plant plankton cannot survive. That's why it's transparent. So you can see a lot of very nice fish without, reduction of transparency. But it could be because of high heat, always evaporation of water is taking place. It can be a good model of climate change when you are interested in. So we are now taking sea samples from Red Sea, then comparing with the sea around Japan. We have not yet result. So, like using ship or just like a kind of buoy, I think such an automatic sequencing for monitoring can be made. Then what kind of microorganisms are there? What I would like to emphasize, approaching to my conclusion, metagenomics is providing us with evolving concept of genomic or genetic diversity, which means, as you know, our cells are made of, if my zero counting is right, then I think 60 trillion cells. However, our God have, I think, 200 trillion bacteria. Our God has a tremendous number of bacteria. And those bacterial composition is related to type 2 diabetes, even cancer, and now it's so much important. Also, we are now trying to collect air. From air, we can isolate DNAs, then we can know what kind of viruses are there. Also, we can have the soil, then we know what kind of microorganisms are there. So tremendous number of different species obtained. Moreover, total number of genes which we speculated will be more than a 10 to the 15th or even 10 to the 20th genes on the earth. Then how come human genes has only 24,000 genes? That will be a big issue, particularly to consider origin of life. So, evolving concept of genetic diversity is a really hot issue, and this is the last. We call biome. The concept of biome, or this is so important to check what kind of microorganisms are there, human gut, or like air, even seawater. So I think biome would be a key term to understanding genomic diversity, particularly new technology called metagenomics. So collaborators, and all DNA should be always world peace. Thank you so much.